Okay, once again, I appreciate all your time. I hope this is helping. Okay, so yeah, really what we're looking at is translation now, the final piece of the central dogma. Translation is protein synthesis. We just briefly talked about that wobble position, that third spot that can vary. Okay, so the thing also what's important to remember about this is the universal codon amino acid chart. In other words, bacteria will use these same codons to make proteins. Human cells will use these sequences of nitrogen bases. These three nitrogen bases make a codon. So it applies for plants, animals, people. Every living thing uses or kind of uses the same code, if you will. So let's kind of talk about what does that really mean to use these three letters to allow an amino acid to make an amino acid. That just is kind of weird. Okay. Remember the difference is though our cells do this, but it happens where they want the protein. Prokaryotes are small. It happens right away. Even as the mRNA is being made, it will make this. So anyway, let's look at what is translation. Okay, so the mRNA will stick to what's called tRNA. So those kind of transcript things. So okay, here's a piece of mRNA, or here's a piece of mRNA. It will stick to tRNA, which here's a model of that. Usually on paper it's kind of drawn flat, but here is where the mRNA is going to stick, right there. Maybe right there, right there. Okay, so anyway, when it sticks to it, it's just naturally attracted to, those nitrogen bases naturally like to stick or bind. So here you can see the RNA is 5' to 3. It's sticking there. So GCC. GCC. You can go to here and find GCC, alanine, right there. So in fact, that is alanine is this amino acid stuck to that. So what's a tRNA? It's a nucleic acid in this funny shape, but at one end, an amino acid molecule is stuck to one end. And the thing is when this RNA sticks to it, this guy, that wobbles, that starts to, when something sticks to something, it changes it. It's going to affect all these side group interactions. This thing does start to move a little bit. It changes shape. It's denatured when that sticks. When this tRNA changes shape, maybe when this guy changes shape, boop, 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 the amino acid pops off. This guy changing shape causes this guy to go out of solution, and then if those pop off in the right order and enough energy is released from all the energy molecules that are in there, and the enzymes floating around in there help create it. But as these amino acids pop off, they bond together and form a protein. The order that they pop off is determined by these tRNAs floating in and sticking to the mRNA, sticking to the mRNA, sticking to it. Pop, 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 go the amino acids. Okay, so here's kind of a fancy way of looking at those steps from your textbook, different textbooks. Quite honestly, let's maybe put this in simpler terms. Okay, so remember, this is the wobble position right there. So not only could GCC make alanine, but also GCU was, GCA, GCG, GC, all of those would make alanine. And so that's the wobble. That spot there can be different things, but alanine would still be your amino acid attached up here. Alanine would still be your amino acid released out into solution. So yeah, so those steps of what I was talking about, the sticking, pop, pop, pop. Here's maybe a simple way to look at it. The amino acid sticks to the appropriate tRNA, then the mRNA sticks to the tRNA. This binding causes the tRNA to change shape. That causes the